If you've done any research on Karen, she, to me, has written more articles on brain activity of the fish than I'm standing in front of. So when I started really looking into it, I couldn't figure out what that meant. Well, I know that when they do brain activities on me, they find nothing. So I figured this lady must know something I don't know. So if you take your time after she speaks today and actually just open the internet, Google her name, you will find more interesting articles that she has written, the research at LSU she's doing now, and then come to find out, it's been since what year? 19 what? That you brought the fish, the original fish in?
Oh, from the 70s. Yeah.
So
I didn't, but someone else did.
The fish population they are researching, they've had in the tanks and bred successfully since the 1970s. So to me, this is a wonderful program. It may lead to other research on other fish. And I tell you what, down the line, maybe they'll be more intelligent than we've ever thought. My fish, I've always said, were more intelligent than me. They know when to wake me up. They know when to tell me to feed them. Well, let me introduce Karen. Karen, come on up. It's a pleasure having you here this morning. Thank you.
Thank you. Not really sure where it's staying.
Wherever you want.
I guess most people are on this side, so if I point on this one, it'll be good, huh? Did everyone hear me?
Yes.
Okay, now? Okay. All right, thank you guys for having me. Today, what I want to do is tell you about some of our research on cichlids that really focuses on how they communicate with each other in different social contexts. And so, just to give you some background on my lab in general, we work on better understanding the sensory and neural basis of different social behaviors like aggression, reproduction, and parental care. And we use this sort of trinity here as a framework for our research, which is loosely organized after the other famous trinity in Louisiana, which is the Cajun cooking trinity here. And so we're interested in the interactions between behavior, sensory neural processing in the brain, and then how environmental signals and an animal's internal physiology might impact the brain and behavior. And so cichlids are obviously great models for looking at how the brain controls behaviors and how sensory information is processed, just because they are so very social. And I don't have to tell that to this audience. You guys have watched them, you know how social they are. But I thought I'd start out with this video. Yeah, well, I can do it either way. So what you want to do, no sound on this one, Fritz. Just watch this fish right here. This is a dominant male. This is his territory. This is just one of the community tanks in our lab. And just watch the different social interactions that he has. I'll try and narrate it as it plays. All right, so here's the dominant male. You can see he has a little border fight with the guy next door. Comes back and chases a few females around. Leaves the area, comes back, goes in the back and has a little fight with the neighbor in the back, comes back, chases some females, goes into his house, does a little cleaning up, spits out some gravel, more chasing of females, another little fight in the back. And so you can see that these guys are just constantly doing these behaviors between aggression, reproduction, and just defending their territories. And so in that quick 30-second clip, that fish had about 10 social interactions, and if you translate that out to the day, it's over 14,000. And then just to put that in perspective, think about how many social interactions the average person has in a day. Anybody have an idea? And not including social media. None? Someone said none? Depending on the study, it's around 5 to 30 interactions per day. So if you're interested in social behavior and how the brain controls that, cichlids are the best model to look at this for. All right, and if you look at the cichlids of the Rift Valley system in Africa in general, they are great models for research because not only are they really pretty and fun to watch, as you all know, but they're extremely diverse. And so they're great systems for looking at things like evolution, speciation, diversity in reproductive strategies and parental care. And so no matter what your research question is, there's probably an African cichlid that's just perfect to address that question. And so my lab focuses on this species down here, Estatotilapia bertoni. This fish is from Lake Tanganyika, but it's also found in the river system surrounding the lake. And we are more interested in this species because it's also great to look at communication, so social communication. So the species lives mostly in these shallow shore pools and river systems of Lake Tanganyika, where the males will set up these sort of clustered territories that they very aggressively defend from rival males, and then they use to court and spawn with females. And the males can be kind of two different types. They're either dominant and territorial, or they're subordinate and non-territorial. The dominant males make up a small percentage of the male population. They very aggressively defend territories from other males. They're really brightly colored, usually with a blue or yellow coloration, and they have this distinct eye bar that signals aggression. They're really reproductively active, and they have very large testes. So they're the ones that are courting and spawning with females. The subordinate males, on the other hand, make up the majority of the adult population. They kind of hang out with the females, so they don't maintain and defend a territory. They're more faded or dull in coloration without that distinct eye bar, and they have really tiny testes, and they're not really reproductively active, so they don't court and spawn with females. But the cool thing about this species, too, is that the males can switch back and forth between these two types depending on what's going on in the social environment. And so they have very different lifestyles depending on whether they're dominant or subordinate. But we're mostly interested in reproduction, and that's kind of what I'm going to talk about today. And so it's the dominant males that are the ones that are reproducing. They have the territory, they're brightly colored, and they're the ones that are trying to get females in there to mate. And so once that dominant male does a lot of courtship behaviors in the water column, which I'll show you a video of that in a second, the females, when they become gravid, it just means they have lots of big giant eggs in their abdomen and kind of get swollen, they get close to spawning. She'll then choose a male and follow him into the territory, lay her eggs on the substrate, turn around and immediately pick them up in her mouth. And then the male comes down and displays his anal fin that has those egg spots on it. The female goes over and sort of nips at those because she thinks they're eggs she still needs to pick up. And that stimulates the male to release sperm directly into her mouth to fertilize it. Then she becomes what's called a mouth-brooding female, and so she broods and cares for those babies inside her mouth for about 2 weeks in this species. And then she'll just release them as little free-swimming fry, and they're kind of on their own. She will take care of them for a few days after the fry release, so if there's a threat, they'll swim back into her mouth for protection. And then after that, she goes through a recovering phase so she can prepare for the next spawning cycle again. All right, so what I want to focus on today is looking at how males and females communicate with each other during reproduction. Okay, so males and females talking to each other, essentially. And cichlids can use lots of different sensory channels. So vision is the most obvious. They're very brightly colored. They do lots of cool behaviors. So we knew vision was important. But historically, a lot of people thought that everything that cichlids did was based on vision because they are so colorful. But a lot of these other sensory systems have been ignored until recently. And so we just wanted to know whether this species was using these other sensory channels during their reproduction. So if you look at social communication just in general, there's always a sender, which in this case is a male, that needs to get some type of information to a receiver, in this case a female. And that can basically provide information on, species identity, what sex they are, what reproductive condition they're in. And then the receiver has to take all of that sensory information, integrate it in their brain with their own nutritional status and hormone state, use that to make some sort of decision, and then that leads to a behavioral response that's appropriate in that context. And oftentimes, the behavior of this receiver can then feed back and influence the behavior and the signaling abilities of the sender. Okay, and cichlids have a lot of sensory systems that they can use. All fishes have these sensory systems. And I'm kind of going to touch on each one of these to show you what we now know about how these different signaling and sensory systems are used during reproduction. And I'm going to start with vision because that one's the most obvious. All right, so we know that visual signaling is important. Okay, so the males are very brightly colored. They also intensify their coloration during courtship, so especially this red patch behind the gills here, and then there's some black markings sort of on their ventral surface, chin area that really intensifies. So there's coloration changes. And then they do all kinds of little flashy displays to try and get females to come in and mate. Okay, so again, here's this dominant male, and he's really excited. So you can see him just swimming out towards the females, shaking his body at them, turning around and kind of slowly wagging his tail and trying to get them to come into his little house. Okay? So males are definitely visually signaling to the females. We also know that females rely on these visual signals to choose a male. Okay, so if you put a male together with a receptive female, pretty soon he starts doing all of those behaviors that you just saw. So he quivers his body, wags his tail, tries to lead the female into the nest, does some digging, and also has coloration changes. The females now can do a number of different responses. They can either decide to follow him into his territory, which means that she's interested and ready for spawning. She can also have no response and essentially just ignore the male. You guys know what I mean there. Or is she going to avoid the male? And that often happens when she's just not receptive or she's mouth brooding. Okay, and she's just like, get away from me. And so one of the big questions we had initially was, how might female reproductive state influence the male's courtship behavior? So in other words, can that male tell when that female is really receptive and ready to spawn? And so, if you look at the female's reproductive biology, when they become gravid, they grow their eggs, they get this nice swollen abdomen, and then they have to ovulate before they spawn. And that just means that the eggs sort of pop out of the ovarian lining and they're ready to be deposited. And so once she ovulates, usually within a few hours, she needs to find a male and spawn with him. So after ovulation, she's sort of on this time clock where she needs to find the male, choose the right male that's really fit, and then go ahead and spawn. And so we wondered whether the males could tell whether a female was ovulated or just gravid. Because he can't see the eggs in there, that's just for graphical purposes. So these are some experiments done by one of my grad students, Julie Butler. And here we did was quantify the number of male reproductive behaviors when he's exposed to either just a gravid female or just an ovulated female. And you can see that when he's exposed to that ovulated female, he does way more courtship behaviors. So the males can tell when the females are ovulated and close to that spawning time. We next focused on the females because we figured, well, if she's on this time clock right here, that's the time when she really needs to pick the right male to choose. And so maybe her vision gets better when she's ovulated and needs to pick up on those visual signals. And so to test that, we basically had a tank split into two with a barrier. We have a courting male on one side, and then our subject females on one side. And we tested both gravid females or ovulated females. And so the females are only looking at the male doing his visual display. So they're only getting visual information, no other sensory signals. We then can basically take their eyes and look at their retinas and see which cells in the retina were activated or turned on when she's watching that male do his visual signal. And so this is an example of a section through the retina, and you can see these two little black dots here are two neurons in the retina that were turned on when she was watching that male across the barrier. And then you can quantify this, and basically what we see is that activation of the retina is much greater when the females are ovulated. Okay, so you can see in this gravid female, there's about 3 cells that were turned on. In the ovulated female, there's a lot more neurons that were activated. And when you actually quantify that with a graph, this is retinal activation in 2 different layers of the retina. But in both cases, the ovulated females have greater sensitivity. So their eyes are just being stimulated a lot more by these visual signals after they've ovulated. right, we looked at this basically the same question using a different technique. And so here we're measuring the actual visual sensitivity of the eye. And so this technique, these ERGs, you can stick an electrode in the female's eye and then flash little LED lights of different wavelengths, and then just look at how their visual system changes or processes that information. And so you can plot that as just, think of this just as visual sensitivity across different wavelengths of light. And what we see is that females, when they're gravid and close to spawning, their retinas are more sensitive, especially at these wavelengths that are sort of in the green-yellow range. Okay, so as they're getting close to spawning, their vision gets better. Then, if you take these same gravid females here, and so this yellow line here is the same one over here. If you take those same females and you inject them with the hormone to make them ovulate, you then see a really further increase in their visual sensitivity. Okay, so as the females are getting close to spawning, their vision gets better, they ovulate, and their vision gets even better. And so what we have is basically a sex-specific and an ovulation-specific change in their visual system that's associated with reproduction. So all of this happens in females, but it doesn't happen in males. So we did a lot of the same experiments in males, and there's no changes. At ovulation in the females, lots of things are happening in the female's body. So there's increases in steroid hormones, there's increases in receptors in the eye itself. The retina gets more sensitive, both to visual cues from the male itself, as well as different colors. And then that may just increase her affiliation towards the male and may allow her to better distinguish certain visual features of the male's courtship so that she can choose which one to spawn with. All right, so that's vision. So visual signaling is definitely important for reproduction. But what about acoustic signaling and hearing? So I'm going to talk about that next. And so a lot of people know this, but when I often give talks to the public, a lot of people don't realize that a lot of fish make sounds. And so all fish can hear, and right now there's 800 plus species of fish, both marine and freshwater examples, that produce sounds. And they can produce sounds in lots of different ways. They can rub pharyngeal jaws together, rub fins and things together, or basically vibrate their swim bladder to make these sounds in the water. And there's only been a couple of cichlids that have been looked at in terms of sound production and how they do it, but the consensus is that it's a compression of the swim bladder mechanism. So here's an x-ray, this is actually a tilapia, but you can see the swim bladder right there outlined, and around the swim bladder is basically the rib cage and some muscles, and so when the body cavity contracts, it vibrates that air-filled swim bladder, and that then produces this little sort of pulse-like sound that goes out into the environment. And then people always are interested in how fish hear because they don't have external ears, obviously, like we do. So it's hard to tell, but they do have an inner ear that's actually structurally very similar to ours. And so inside that ear, there's a sensory area that has hair cells on it, and above that is a dense otolith that's made of calcium carbonate. And so the way fish hear is that as a sound wave comes at the fish, basically the entire fish moves a little bit with the sound wave back and forth. And I gotta play this animation. Yeah, so if you look at the sensory hair cells, and here's the otolith on top of it, what happens is because this otolith is much denser than the fish tissue, its movement lags behind the hair cells a little bit, and so that creates this shearing force that stimulates the hair cells and that gets sent to the brain as neural impulses that the fish then perceives as sound. All right, so the first thing we needed to know was, well, are these males actually making sounds during reproduction? And so this is what we're making sounds. This is just a simple experiment. Here's a hydrophone, which is just an underwater microphone in the tank. Here's the dominant male. He's the one that's going to he's going to produce two separate courtship sounds here. And then there's some receptive females in there. first little grunt sound. And that's the second one. So you can see the male is doing this when he sort of shakes his body and presents it sort of at the female. And so these sounds are relatively low intensity, and they're usually only produced when the male is quivering his body in close proximity to the female, so it's probably a close range communication system. The sounds are pulsed, so here's what the waveform looks like, and then this is a sonogram that just shows the frequency components of the sound. So the pulses are 2 to 19, there's an average about 7 or 8 per sound. They're 50 to 700 milliseconds long, and the peak frequencies are around 300 to 700 hertz, but they're really broadband. They have very low frequency components, and they go all the way up to 1200 hertz. We also know that the frequency of the sounds depends on male body size. So actually large males produce low frequency sounds, and small males produce high frequency sounds. And that's really interesting because it means that the females can use frequency of the sound as an honest indicator of male body size. So the female just listening to a sound can sort of tell how big that male is. Yeah, we can't do that. Oops. All right, and if you look at sort of a time sequence of when they're doing these sounds, so all the sounds are produced when the male quivers his body, but not every quiver has a sound associated with it. And that's important because it means that the sounds are not, that the sounds are intentional. So it's not just a byproduct of the body movement, they can control when they make the sound and when they don't. And so if you just look at, this is one trial from one male, so each of these white ticks is when he's doing that visual body quiver, and then the red is the ones where he's making a sound. But you can notice that there's areas here where he's doing a body quiver, which is a visual signal, but there's no sound production. And so this male, about 40% of his quivers had sounds associated with it. And then here's another male that had 80% of his quivers had sounds. And we also notice that this percentage is actually related to male body size too. So larger males tended to have a higher percentage. And we still don't know why that is, but it may be because the males that are larger, they're older, they're more experienced, and they're better able to tell when a female is really receptive because they've done lots of courtship before. And so that they can only do their costly sound production when they're sure that that female's ready to spawn. Okay, so males are making sounds, but do the females care about them? And so the next experiment we did was we did a playback experiment where we put an underwater speaker in a tank and we could play male courtship sounds out of that speaker. And then we measured how much time a receptive female spent hanging out with a male that had sounds coming from his territory or a male that had no sounds coming from its territory. And what we found is that when you play courtship sounds through that speaker, that female definitely prefers to hang out with the male that's making sounds. When you play just a noise control sound through the speaker, she shows no preference at all. So it's not just any noise coming out of a speaker, she's specifically attracted to the courtship sounds produced by male. Okay, so the females do care about the sounds. So when we saw this, we wondered, well, if she's using sounds to pick out males during courtship, maybe her hearing also gets better or changes with her reproductive cycle, just like we saw in the visual system. And so we tested that by doing this technique called auditory evoked potentials, which is very similar to an EEG in a person where you put electrodes on someone's head and you measure the brainwave responses. Apparently some of you have no brainwave responses, but the fish do. But in our case, the electrodes are little needles that you just sort of stick underneath the skin above the brain case. And then we have an underwater speaker here, so the fish is here, underwater speaker. We can play sounds at different frequencies and intensities and then measure the brainwave response. And so here's an example of a typical brainwave response. So this is the stimulus coming through the speaker, so say a 200 hertz sound, and then an increasing threshold, so a very loud sound to a very quiet sound. And then from this, what we can do is basically determine a threshold for each frequency. And so the threshold is just the lowest sound intensity that gives us a measurable brain response based on our criteria. And then you can plot these thresholds across every frequency, and you get what's called a tuning curve. Okay, so some people have probably had this done to test their hearing, right? So this is hearing thresholds on this side across different frequencies, and we measured the hearing in gravid females, which are getting ready to spawn, that's in the red, and then mouth-brooding females, which are in the yellow, which shouldn't really care about what the males are doing because they're not ready to reproduce. And what we see is that these gravid females have 2 to 5 folds better hearing than the mouth-brooding females. And so lower threshold means better sensitivity or better hearing. And it's most dramatic here at these very low frequencies from 200 to about 400 hertz. And those are the same frequencies that are contained in the sounds that the males produce. Okay, so as she's getting close to spawning, her hearing is getting better, specifically in the frequency range of the male sounds. And this type of hearing change and sensitivity it's not only unique to this cichlid, it happens in other fishes, birds, and it even happens in people. And so women, especially going through their menstrual cycle, have changes in their hearing sensitivity, as well as premenopausal and postmenopausal women. And it's all due to circulating steroid hormones, so especially estrogens or estradiol. And so we next just wondered, well, is the better hearing in these females also related to hormones? And the really quick answer is yes, it is. So if you just plot fish hearing threshold across their circulating estradiol concentrations in the bloodstream, we see that females that have higher estradiol also have better hearing or lower thresholds. And it's not the case if you look at androgen hormones. This is just 11-ketotestosterone, which is a fish-specific androgen. And so what's happening here is gravid females, they're growing their eggs and producing more estradiol. That's somehow translating into improved hearing. We don't exactly know where that's happening yet. And that may allow them to make better mate choice decisions based on the sounds produced by the male. All right, so we know that hearing is also important. But we also wondered, well, if the male is shaking his body and producing the sound, is it giving the female the same information or something different in those 2 different sensory channels? So another one of my grad students, Tisha, is doing this experiment to look at that. And in this setup, we have a courting male on one side of the tank, and then our subject, receptive, gravid female on the other side. And then by changing the type of barrier between the 2 of them, we can allow that female to get visual signals only from the male, sound cues only from the male, or combined visual and sound cues to see how her behavior changes. And so if you look at, first we're going to look at the male's behavior in this context, just as the number of quivers in the association zone, which is just close to the barrier, meaning that he's directing it towards the female on the other side, across the different sensory conditions. Basically, there's no difference. So that's what we wanted to see, because we wanted to make sure that this male was giving the same reproductive effort across different trials. We're just controlling what senses the female can use. Okay, we're telling her what senses she can use. And then if you look at the female behavior, just as time in the association zone, so again, whether she's interested in the male or not. And here we see that she spends a lot less time over at that barrier when she can only hear the male. Okay, so she really needs those visual signals in order to be interested. So sound alone is not enough. But this pattern of activation tells us, based on the literature, that the visual and acoustic channels are non-redundant. So she's getting different information from both of those channels. It's not the same thing. But the vision actually dominates. And that's the case with all the sensory stuff we've looked at. Vision dominates, but the other senses provide some additional information. All right, so we're also interested in where the brain is processing this information. And so we can take the females from those same experiments when they're behaving in those different sensory conditions, take out their brains, section them, and then use different markers to label neurons in the brain that were actually firing when she was in those behavioral contexts. So these purple dots here are all individual neurons in the brain, that were turned on when she was in those behavior contests. And I'm not going to show you a ton of data for this because it gets tedious, but I just want to show you an example of what types of things you can get out of this. So here's a brain section. So all these little purple dots are neurons that were activated during those behaviors. When she only hears the male or when she only sees the male. And you can't really see much difference. There's some neurons that are turned on, but it doesn't look any different. But when she can see and hear the male, you see lots more neurons activated. So she's getting different information in these two channels, but when they're combined, they're really important and influence her behavior even more. Another thing we're doing to look at this is we can actually stick electrodes in different regions of the brain and look at what a single neuron is doing at any one time. And so I'll just play you this quick little video of a recording. So basically this line right here is going to be a neuron that's firing action potentials. And so every little vertical tick mark is a neuron that's firing. And then we're going to play a sound to it, and you'll see how the neuron changes its firing. Actually, I do need sound for this one. Because you can hear the neuron firing as well. So this is just spontaneous activity. A lot of these neurons are just active on their own. And now we've turned on the sound. And you can hear the neuron is basically firing in time with that sound. So this is how these fish get information about the frequency and the intensity of sound in their environment. But we can look at what's going on at a single neuron level, and then we can do things like manipulate hormone levels and see how it changes their firing properties. So we're just starting these experiments. All right, so hearing is also important. I want to briefly touch on the lateral line system. So all fish have a lateral line that they use to detect water movements in the environment. And so we thought that while these males are quivering their body, they're giving a visual signal, they're making a sound, but they're also basically pushing water at the female. So is the female using this water movement for reproduction as well? And so the lateral line system in fish is a series of receptors that are found either on the skin or in canals beneath the skin. And so it's a little hard to see here, but all these little green dots are a functional unit of the lateral line system over here. And each one of those is called a neuromast. And it's basically a bunch of hair cells, so similar to the inner ear, that's covered by this gelatinous cupula. And when a water movement comes past the fish, it basically tugs on this cupula, deflects these hair cells, and that sends neural impulses into the brain about what that water movement was. All right, so to look at whether the females were using water movement signals during reproduction, we did an experiment. These were done by a couple of students of mine, Julie and Chase. So if we put a male and a female together in a tank, what we measured here was the female follow response. So if a female is interested in a courting male, she will follow him and go into his territory, and eventually that will lead to spawning. Okay? We put in females that either had an intact lateral line so they could sense water movements, or females that had their lateral line ablated, which means they couldn't detect any water movements. And so this is the percent of courtship attempts done by the male. And so when the female has her lateral line intact, she tends to follow him into his territory. But when she can't feel those water movements and has her lateral line ablated, the amount of following she does is reduced. So it's negatively impacting her ability to, or her motivation to spawn. We also noticed that when we had females' lateral lines intact, about 70% of those trials ended in spawning, so it went all the way through the sequence. But when she couldn't feel those water movements, only about 30% of those trials ended in spawning. So this tells us that the lateral line is also used by the females during reproduction. All right, that's all we know about lateral line right now. Olfaction is a really, really important sense in a lot of fishes for courtship and reproduction. And the cichlid or general fish olfactory system is an olfactory epithelium that's beneath a nares opening on either side of the head.
this is what the epithelium looks like. It's kind of a rosette or flower shape in most cichlids. And then on each of these folds, you have a bunch of olfactory receptor neurons that will bind different odorants in the water column, and that will send neural signals into the brain for processing. And so the first question here is obviously, well, does this species use chemical signaling during reproduction? And one of the ways that fish will chemically signal to each other is through urination. Okay, so I will be talking about fish pee for the next few minutes. And what we can do to visualize their urination is you can inject them with an innocuous sort of blue dye that concentrates in the urine, and then you can see it as these little blue pulses that are released every time the fish pees. And so you can look at how much they pee in different contexts. So these are experiments done by another grad student, Karen Field. And so to look at whether they were signaling to each other, we had a tank where we had a dye-injected subject fish. It was either a male or a female. And then we could expose them across a barrier to different types of stimulus fish and then see how much they urinate. So for dominant males, we exposed him to gravid females that were getting ready to spawn, breeding females that shouldn't really be interested in courtship, and then just a no-fish control. And then to see if females were signaling back, we had a dominant courting male or a no-fish control. So we'll look at the males first. So this is just the percent change in urination rate of the males across these different conditions. And the bottom line is that he really increases his urination activity when he's exposed to that gravid female that's close to spawning. So he's specifically peeing at the females that he knows are ready to mate. And then another interesting thing is I did a couple experiments where I tried to look at visual signaling, sound production, and urination from males all at the same time. And it was really interesting that the urination didn't happen at the same time as those quivers. It was always in pauses. So the males would go up and quiver and make their sound production, they would pause, pee, and then go back to their courtship. Another interesting thing we noted is that a lot of the urination tended to happen sort of early on in the courtship sequence, so that they would pee in the beginning, but then once they really started vigorously doing their quiver activities, they kind of stopped urinating. So the idea is that maybe they're releasing some chemical that's stimulating the female either to ovulate or to do some receptive behaviors to signal that he's ready to go and they can coordinate their spawn. All right, so the males are peeing to the females, or the females peeing back. Of course they are, so this is urination rate of the females when she's exposed to nothing or a courting male, and she definitely increases her urination when she's exposed to a male that's courted. So what we have here is that both males and females are signaling to each other with chemicals during reproduction. So one of our next questions was, well, how might these chemosensory signals from the females actually influence males' behavior? And so to test this, we had a subject male fish, so we're going to quantify his behavior in the center of the tank. And then we can expose him to a gravid female that he can only see, that he can only smell by just delivering chemicals through that tube, or if he can see and smell that female at the same time. And so here, this is just a number of reproductive behaviors. So here's basically what happens. When he can't see a female and he can't smell a female, he doesn't do any courtship behavior. Okay? He has nowhere to direct it. When he can see the female but not smell her, he does start to do some courtship. So vision alone is enough to get him motivated to start doing courtship. When he can't see a female but he only smells her, again, he doesn't do any courtship behaviors. But that condition is really interesting because while he's not courting, he's basically swimming frantically up and down in the tank, trying to figure out where that female smell is coming from. But he can't see anyone, so he doesn't know where to direct his behaviors. But when he can see and smell that female, he dramatically increases his courtship. So both of these channels are important. Chemosensory information is providing some crucial information that he's not getting from the vision. And so again, this is an example of non-redundant signaling where they're getting two different messages from each signal. All right, just related to olfactory stuff, we also know that males will pee at each other, especially during aggressive contexts. And so we wonder here whether, since we have dominant and subordinate males and they can transition back and forth, they have very different lifestyles, we wondered whether their olfactory systems might be different to basically help them with their status-specific lifestyles. And so, for example, the dominant males are really focused on reproducing and defending their territory, whereas the subordinate males don't do any reproduction, they're not aggressive at all, they're basically fleeing from the dominant males all the time. And so they're very suppressed. So their needs are very different. So the way we tested this was to do some single neuron recordings in olfactory regions of the brain. And so these are some experiments done by a postdoc of mine, Alex. And what we did here was we can pass odors across the olfactory epithelium. So we tested a whole bunch of different things. Female water and male water are just, female water is water that had housed behaving females. So anything they're releasing in their skin, their gills, or through urination is contained in that water sample. Male water is the same thing, it's just behaving males. This is a steroid that's thought to be a pheromone in some species, a food-related odor, and then just some controls. And then while we're doing that, we have an electrode in the olfactory center of the brain, and we can record the neural impulses. All right, I just want to show you an example of a neuron recorded from the brain of a dominant male and one from a subordinate male to each of these three different odors. So this is when the stimulus is passing over the nose, and so each of these vertical ticks is when the neuron fires. And so what we see for the male water is the dominant males have a little bit of a response, but it's not that dramatic. The subordinate males respond really well to the male odors. But if we look at the female water and the steroid, we see the total opposite. So the dominant males are really sensitive to female water and the steroid, whereas most of the subordinate males didn't respond at all to those odors. And so this is really interesting because it means that their forebrain olfactory system may be very specific for their lifestyles at that particular status. So if you look at what they have to do, these dominant males here, again, they're defending territories, they're courting, so they really need to pay attention to female-released odors because they need to find mates and spawn as much as they can. The subordinate males, on the other hand, are doing submissive things, they're hanging out with the females, they don't have a territory, but what they are doing is monitoring opportunities to rise in rank. So they may be using olfactory signals released by other males in the population to tell which males are dominant. So they know when to be submissive and when they might want to challenge a weaker one to possibly gain his territory and take over. So this was a pretty exciting study to us. All right, taste. I want to spend a couple seconds on taste really because there's no evidence really in any fish species that taste is used for reproduction, but it's obviously very important for things like feeding. And then there's lots of different types of taste buds that are found in fish, both in the mouth as well as sort of all over the body, the fins, the gill area, etc. And so all I want to do is show you this really interesting video that we did a couple months ago. This is in collaboration with John Caprio at LSU. And just to show you how strong the feeding response is in these guys if you stimulate their taste system. So there's going to be two videos here playing on top of each other. It's actually the same fish. You want to watch sort of right about here in each tank. We have a tube underneath the gravel that we're going to pump in either just water in the control situation or this secret sauce, which is a proprietary mix of amino acids and other things that people won't tell us what they are. So we're just calling it secret sauce. And there's also a blue dye in there, which the fish don't react to, but so you can see the when it comes out. And it's actually sped up in time too, so it's not real time. But if you start to watch, you can see the blue starting to come out here in both cases. These guys are pretty much ignoring it, but you start to see this really strong feeding response. I was super surprised to see how strong this was, but basically you're triggering their taste system, which is partially reflexive, and they're just going to town there. Okay, so kind of what I've shown you today is that we now know that courtship and reproduction in this species, and I'm guessing a lot of other cichlid species, is very multimodal, so it's not just vision. Okay, we have evidence that all of these senses are used during courtship to different extents, and that each sense really provides different information to the receiver, whether it's a male or a female. We also know that different sensory information may be important on different spatial or temporal scales. So for example, one sense might be important far away, and then other ones are more important close by. Different sequences, some early on in courtship, some later. And also in terms of noise in the environment. So for example, if the visual conditions are poor and it's really turbid, which you do often get in those shallow shore pools, hippos walk through there all the time in the wild and can totally mess things up. So if they don't have vision, then they can use some of these other senses. Okay? And then lastly is that we have a lot of evidence basically that sensory perception in these guys changes with reproductive condition or with male social status. So we have examples in males and females for multiple sensory systems. And so this is probably a lot more widespread than we thought. It just hasn't been investigated in really any other species. And then I just want to spend how much time do I have? Am I over already? All right. I just want to spend a couple minutes telling you about some other work that we're doing that's basically using cichlids as models for biomedicine. So I'm definitely more interested in the fish itself because it's a fish and I've always loved fish and they're cool. But sometimes we need to convince funding agencies that we're doing something that's related to humans. And so that's why we have a couple of lines of research where we're doing that. And so I just want to give you an example of what we can use these fish for. And the reason is that fish are vertebrates, and so a lot of their brain structures and sensory processing are very similar across all vertebrates, including in humans. And so by doing just basic research in a fish, we can provide insights to translational research, even in terms of developing therapeutics in humans. And so by doing this approach, we can both uncover things like species-specific adaptations, so things that may be unique to one or a few different fish species, or identify conserved neural mechanisms that are there in fish and they're also there in us. And so there's two lines of research that we're kind of exploring this with. One is the neural control of maternal care and feeding, looking at trade-offs between feeding and offspring care. And then the second is as models for neurological conditions in people. So in terms of the maternal care stuff, maternal care is very important across all vertebrate taxa, including in people. And there's often trade-offs with a mother. She needs to sometimes decide whether she's going to feed herself or feed her offspring, or protect herself or protect her offspring, right? So how the brain is controlling those motivational switches is really not understood in any animal that well. And so these mouth-breeding species have a really extreme form of parental care, right? So the mouth-breeding females, they're starving themselves, but they're taking care of the babies. Whereas when they're not mouth-breeding, they're gravid, they're doing lots of feeding, and they're not taking care. So it's basically offspring care here and self-care here. But how is the brain controlling all of this? And if we can figure out and identify some of these mechanisms, it has really important implications for things like anorexia and metabolic disorders in people, because a lot of the molecules in the brain regions are very similar. And then also in terms of maternal care, we're just interested in how the brain allows a female to go between these drastic feeding states and also these drastic maternal care states. So no care to brood care inside the mouth, and then she will take care of those fry for a few days afterwards. So when does she decide to stop doing that? And sometimes will eat her own babies. This is just, I'm sure you have all seen this, but this is just a little video of a female taking her brood back in after they're threatened for protection to her mouth. So we're looking at lots of brain activation stuff with these behaviors. And then the last thing is using cichlids for neurological disorders. And these are some experiments that my PhD student Rose is starting right now. And we're using this social defeat stress paradigm, which we can then mimic the scenario that happens in bullying, stress coping, and also PTSD. So it's a typical resident intruder paradigm. If you have two dominant males to start with, the resident is a little bit bigger, the intruder is a little bit smaller. And then if you move that intruder into the resident's territory, there's a little fight, and basically the intruder almost always loses, starts changing his color to become subordinate and become submissive. And then if you repeat this over successive days, it's basically like, you know, going to the playground every day and getting beat up by the big kid. Okay, so this is directly related to bullying. And then we can look at their brain and behavior and see how it's changing or modified. And so one thing she's doing right now is really trying to set up a PTSD model. And to do that, we are using 3D printed cichlids. So we can print out cichlids in different shapes and sizes, and then she paints them. And so this one is a dominant male. And then we have this little swimming apparatus that we can make the fish just sort of swim around in circles in a tank. And so this way, there's also a real fish in there, and you'll see him come over and attack it at one point. But now this fish is not being aggressive, but it's still a dominant male. Okay, so we can control the aggression level by using these models. And the idea for PTSD is you do this repeated social defeat for a period of time, then you wait some period of time, and then you reintroduce the fish to this model and see if they behave like they did when they were originally bullied, or if they adopted a new strategy. So it kind of mimics, you know, if you're off at war, saw some really horrific things, went home for a time, and then there's some trigger that gives you back the feelings and the symptoms of that event. All right, and that's where I want to stop. So I'd like to thank my lab. I've had lots of great students, grad students, undergrads that have helped out with this research over the years, our funding agencies. I want to thank the organizers here and the sponsors for allowing me to come and speak with you. You can check us out on Twitter if you want and see what kind of research we're doing. And then lastly, I just want to make a plug for our blog if you haven't seen it. It's called Bert's Blog. Here's the address here. And this is basically an outreach thing for us to get our research out to the community in a fun and easy to understand way. And so basically it's told by Bert, who's a dominant male, and his girlfriend Tony. And then there's other characters like Tyrone that come in. But we tell stories about our science and our results from the fish's perspective. So basically Bert and Tony are telling stories about how we're using them for research and what we're discovering. So you can go check that out, it's kind of fun. And with that, I will stop and take questions now or later on if you have them. Thank you very much for your attention.